Hello and welcome to the TissueGnostics Academy, a webinar series where we ask researchers in our network to present their research, methods, and publications. My name is Carly Briffa, and in this video, we are joined by our guest, Dr. Nicolas Hoch, from the Department of Biochemistry at the University of Sao Paulo, to present his research on quantitative image-based cytometry with a focus on the foci. So when you're ready, Nicolas, you're welcome to start.
Thank you. Thank you very much, and for the opportunity to be here and share a little bit about the sorts of analyses that we're using our TissueFAXS system for. So my lab is interested in how cells signal and repair DNA damage. And so what happens is when we use cells in culture, growing in a 2D layer like this, and we stain for markers that mark DNA damage sites, such as histone H2AX, which gets phosphorylated in response to mainly the DNA double-strand breaks. What you see is that there is a huge variety in the intensity of this signal between cells. And so this heterogeneity is very important in order for us to actually understand the biology of how DNA damage is signaled and repaired. There's a strong link between DNA repair and the cell cycle. And so it's very important, as you can see here, you get a big variety between the signal within different cells in the population. You can have cells where there's basically no DNA damage, and cells right next to it where there is a lot of DNA damage. And often this is caused by differences in the cell cycle stage these cells are in. And so it's very important for us to actually analyze populations of cells that are in the same stage of the cell cycle in order to understand better how these signaling and how these repair events are happening. And so, as I said, we work with cells in culture that are growing in a 2D layer. And so we seed these cells on glass cover slips, and then we can prepare slides where multiple cover slips are prepared like this. You can then use the TissueFAXS system to preview your slide, and you can see these different cover slips here. You can then choose areas that you want to acquire. You can remove areas where cells might have detached a little. You can avoid areas where cells have concentrated too much. And so we tend to choose areas that are somewhat more sparse, so we can separate the nuclei more cleanly. Often we will stain cells with multiple markers. So we have 2, 3, sometimes 4 colors. And so you set all your acquisition parameters, you set the autofocus on the system, and then the software will, and the microscope will acquire this whole area that has been selected here, taking then images in these different channels for each of these fields of view. So in this case here, our area that we chose is comprised of 100 fields of view, 10 by 10, and we take the images then at, in this case, a 40x magnification. And so all of the acquisition of those images is then automated, and the software stitches these images together so that we end up with essentially one big image of hundreds or thousands of cells that we can now analyze. And that's when the interesting work then starts. So we stain cells with DAPI, which is a marker that will stain the DNA and will stain the nuclei. So here is just the DAPI channel of a very small area. You have 5 nuclei here. And the first challenge for the software is to identify these nuclei as cleanly as possible. And so we do a couple of operations here to help the software identify these nuclei more cleanly. The first step we use is something we call unsharp mask, which is an operation that will essentially remove the background. So although this image looks black wherever there are no nuclei, there is actually residual signal here that we remove with this operation. And essentially now the background is 0, the pixel intensity, the signal intensity is 0. It does remove a little bit of the true signal, but what we do next is then convert this 16-bit image into an 8-bit image. And in that process, we also saturate the signal in a way that now the whole background is completely black. And now wherever there was a nucleus, the signal is now essentially completely white. And so this is a much cleaner boundary for the software now to try and detect where the nuclei are. And so then the nuclear detection algorithm then identifies these edges and then we can map back this detected nucleus onto the original image. And this is important. We always have to use the original image to identify and quantify things. We can use the manipulated images to help the software get to what we want to detect, but we always have to go back to the original image to quantify things from there. And so we then use this this detection of these nuclei, and we can then ask the different questions about those nuclei. One of the simplest experiments we do is to quantify cell death. So in this case, instead of fixing cells and then staining them with DAPI, we use live cells and we stain them with this other DNA dye called Hoch. Hoch will enter the cell and will enter the nucleus and stain all of the nuclei in blue. So you can see here that all of the cells are stained, all of the nuclei are stained. We use a second dye that also stains DNA called propylium iodide, but this marker will only enter cells that are dead, where the membrane is ruptured because it cannot cross the cell membrane. And so only dead cells will be stained with PI. So now you can just quite simply quantify the percentage of cells that are, the percentage of nuclei that we detected with the Hoch channel, what percentage of those is also PI positive. And so then you can quite simply here, using microscopy images rather than flow cytometry, for instance, you can quantify the percentage of dead cells in your population. Now, often what we want to do is a bit more complex than that, and we actually have to select and identify these nuclei a bit more carefully. And so often what we do is a two-step mechanism here in order to select the correctly identified and correctly detected nuclei. So one thing a correctly identified nucleus should have is that it should have an expected area, an object that is too small or too big to be a single nucleus, we want to exclude. So this is on this axis here, the DAPI area. We also use another measure called DAPI compactness, which is essentially how round that nucleus is. So objects that are not particularly round will be down here, and those will be often, an incompletely detected nucleus, or sometimes two nuclei next to each other that the software identified as a single one. And so we gate this population of cells here that we're interested in that has an expected size and an expected roundness, if you like. And we further analyze the cells within this gate in a second scatter plot here, where we take the area, so again, the size of the nucleus, and now we use the intensity, the total sum intensity of that nucleus. The DAPI stain is binds to DNA, so the intensity, the total DAPI intensity within the nucleus is proportional to the amount of DNA within that cell. And so a cell that hasn't been completely identified, or a nucleus that hasn't been completely and correctly identified, will have lower DNA content than expected, and doublet events or cells that are bigger than expected will also have more DNA than expected, and we gate those out using this gate here. You can also observe that there are already two populations of cells here. What happens is that cells that are in G1 have a certain amount of DNA, and their nucleus tends to be slightly smaller. As cells progress through the cell cycle and make a copy of their genome, they actually have double the DNA in G2. And so in G2 nucleus will have more DNA content. In theory, double the DNA content as a G1 cell. And the nucleus also tends to be slightly bigger. So this population of cells tends to have a bigger DAPI area, as you can see here. And so then we gate, in this two-step process, we gate out the nuclei that we trust are correctly identified, correctly detected, and have the expected DNA content. And after that, we can start separating cells into cell cycle stages. And to do that, and to improve on that, what I just showed you, we also use an additional stain where we treat the cells with a thymidine analog called EDU. And EDU gets incorporated into the DNA. The DNA polymerases think it's a thymidine, so it gets incorporated normally. And then this EDU that's been incorporated can then be covalently modified with a fluorophore, so we can detect the cells that have incorporated EDU into the DNA. We do this EDU treatment for half an hour, so only cells that have been actively replicating within that half an hour will incorporate this EDU. And so you can see here, for instance, 4 nuclei, 2 of which were in S phase, were replicating their DNA within that half-an-hour window. And these 2 other nuclei have no EDU signal, so they were not replicating within that time frame. And when we now plot the cells based on the DEPI intensity and the EDU intensity, again, you get cells down here that have less DNA, which are in G1, and then cells that are down there that have more DNA that we expect are cells in G2. But you can see that with the addition of this EDU stain, we can separate these populations much cleaner and identify the cells that are in S phase that are replicating. What happens is that cells in G1 have a certain DNA content, and they are EDU negative. But cells that are entering S phase incorporate EDU within this half-an-hour window, but their DNA content is still essentially the same as a G1 cell. They haven't had time to make enough DNA for this to actually affect the DEPI signal intensity. So these are the cells that are in early S phase. They have a smaller amount of active replication forks within that half an hour and are incorporating less EDU on the left-hand side here. Cells that are sort of midway through replicating their genome will have incorporated more EDU within this half-an-hour window. There are more ongoing replication parts at the same time, so more EDU gets incorporated within that half an hour. And the DNA content starts shifting to the right because the cells are making new DNA, right? So that the DEPI intensity increases. As the cells reach the end of S phase, the amount of EDU incorporation within that half-an-hour time frame goes down again because there are less active replication forks. And the DNA content is now closer to a G2 cell. And so then they will gather around this side of this graph. And so you can, if you want to, you can gate cells into early, mid, and late S phase. But what we tend to do is to gate cells into G1, S generically, and then G2. And so with these sorts of graphs, you can now quite easily determine changes in cell cycle profiles, for instance, between different treatment conditions. So, you know, our untreated population is like this, and then you treat your cells with something that damages DNA or whatever, and you get, in this case here, an accumulation of cells in S phase, or in this case here, we had an accumulation of cells predominantly in G1. But something a bit more interesting that we're able to do with this technology now is to start gating out these populations of cells that are in G1, S, and G2, and then asking for, you know, the intensity of other markers that we might be interested in. So this is an example of an experiment where in the untreated situation, we had a certain amount of marker X here, the biology doesn't matter, and then we treated cells with agent A. And we saw an increase in this marker in the cells that were treated with agent A. But we also saw an increase in the same marker when we treated cells with B and C. And when we averaged out, you know, over the whole population of the cells, the induction by A and B and C seemed to be the same, okay? But when we gated out the cells into different populations, we noticed that treatment A was actually inducing this marker in cells in G1, whereas treatments B and C were inducing the same marker in cells that are actually in G2. So this suggests that, you know, if we hadn't separated these cell cycle populations, we would just say, oh, look, A, B, and C seem to be doing the same thing. But biologically, what's happening here is completely different, right? We get induction of this marker in different stages of the cell cycle. So there's new biology for us to understand here, and we're working on that at the moment. In the process of this project, we also started wondering about this population of cells that we are calling G2. So the way we're gating out these G2 cells, we actually have, we're just assuming that the DNA content of those G2 cells is double a G1 cell, and they're not incorporating EDU, so they cannot be an S. But we know that in tissue culture, cells at a perhaps quite low rate, but there is a propensity for cells to become tetraploid in culture. And that depends a little bit on how normal your cell culture, your cell line is. But this does happen in culture. And so what we wanted to be able to do is to separate the cells that we are calling G2 into the ones that are truly G2 from the ones that have become tetraploid and have double the DNA content as a normal cell, but are actually in G1. they would fall in that graph that I showed you before, they would fall into the same area. And so we used another stain called geminin. And geminin is a marker that is a protein that gets degraded in G1. And as cells enter S phase, they re-express geminin, and geminin levels remain high in S and then in G2. So again, by DEPI DNA content, you'd say this is a G1 cell, and then everything here is a G2 cell. But actually, based on geminin staining, we can say these cells up here that are geminin positive, those are true G2 cells. These cells down here, they have a G2-like DNA content, but they don't express geminin. So they must be tetraploid cells that are in G1. And if you'd allow them to go into the cell cycle, they would actually form another dot over here somewhere when they are in G2. Okay, so this way we can actually separate not only cell cycle stages, but also diploid cells from tetraploids. Another something that we've started doing a lot lately is to plot our data in what we call superplots based on this publication here. Very interesting. So as I showed you, what we're doing is we're acquiring thousands of cells per sample. And then we average out the signal over these 1,000, 2,000 cells, and that becomes our one replicate experiment. And so we take the average for N1, N2, and N3, and we plot them, and we just plot, the average of the 3 replicate experiments. But when you do that, you actually lose the richness of information that went into generating these averages. And so you cannot use individual cells for each experiment as independent ends, right? We couldn't say we have 6,000 N for this experiment, because that's not what we did. But you can show the underlying data in your final graph using this, what they call superplot, which we find very useful to show the heterogeneity that's actually happening in the tissue culture dish. So each of these, green circles or blue squares or whatever is a replicate experiment, and this is the average for that replicate. We can then still plot an average between the 3 and standard deviation or whatever, but we plot the individual cells that went into these averages as these gray dots in the background here, and that shows you the heterogeneity that was present in that population. So that's a nice way of plotting the data and showing the richness of information that went into that. Now, the experiments I showed you so far were based on just quantifying the total intensity signal within the nuclei. But I showed you at the beginning that H2AX and some other DNA damage and repair markers will actually form what we call foci. And so foci are these structures where a protein or a marker that is generally dispersed throughout the nucleus, like this nucleus over here, in response to DNA damage, will localize to the DNA damage site and will form these foci. And so this is a marker 53BP1 that forms these foci within the nucleus. And what we're interested in is not quantifying the signal intensity of 53BP1 per nucleus. We want to know how many dots there are and how big they are. And so for that, we need to detect the nuclei, but we then also have to detect these individual foci. And using a similar strategy to what I showed you before, you can determine the nuclear boundaries for each of these nuclei, and you can also detect where these foci are. And then, based on that, extract the number of foci per nucleus, or the total signal intensity contained within these foci for each of these nuclei, and then extract biological information from that. Something slightly more complicated that we've done in a publication that we did published late last year was when we were studying something a bit unrelated. We were looking at coronaviruses and a function of a protein that's contained in coronaviruses. But the point here was we had a signal that formed essentially foci, but these foci were not within the nucleus. So this was a cytoplasmic structure, some sort of dotted structure in the cytoplasm that we wanted to quantify. And so what we did was we determined the boundaries of the nuclei again, and then we grew a ring around the edges of this nucleus. which was a 30 micron ring. If two cells were too close to each other, then this ring would stop growing, and you essentially get a boundary between the two nuclei, somewhere roughly in between. I mean, we don't have a true marker for the cell cytoplasm here. We're still working on it. It's actually harder than it sounds to get a nice stain for the cytoplasm to do that. And so the other thing we did then was so we grew this ring. We stopped the ring from growing if it touches the neighboring cell. But we also allowed this mask to grow further if there was still signal, in order not to lose any signal that might be a bit further away from the nucleus than expected. And so as long as there is signal, this boundary was allowed to grow. Okay, in this way, we could then detect again these foci in the cytoplasm within these cells, quantify how much signal was contained in these foci, and then see the effect of our treatments on that. So again, here you see the variation between cells, but it's a very reproducible induction over several thousand cells here, and you can plot that data in the graph. And so this is the sort of stuff that we were doing in my lab. We have a bunch of people working on some very interesting projects. As I said, we're based at the University of Sao Paulo here in Brazil. We're always open to receiving new students and new fellows, so please get in touch if you're interested in this work. We are funded by several agencies here in Brazil, and they're often quite happy to have international students as well, so please get in touch. Thank you very much, and see you later.